Topic of my lecture is Introduction to Bioinformatics, and I am Sumati Hajela, Associate Professor, Department of Biotechnology, IPS Academy. So, Introduction of Bioinformatics. Bioinformatics word may be split into two parts, biology and informatics. So, informatic word says something about computer science and information technology, while bio word says about biology. And these both disciplines merged into a single discipline, new discipline, which is known as bioinformatics. So, bioinformatics is a science which utilizes computer science and information technology to manage and analyze biological data. Bioinformatics has grown with a very fast pace and evolved with the initiation, execution, and completion of human genome sequencing project. So, starting from early 1990s, we lived in pre-genome era when our genome sequence was not known, and after 2003 onwards, after 2003 and beyond that, with the completion of Human Genome Project, we are already living in post-genome era. Biologists used to face certain problems before that, and that were handling storage and management of biological data, along with its understanding and comparative study. Uh, This says that quantity of information was large, and handling and keeping, storing that was difficult in the case of biological data. And when they cannot be handled or stored, their comparative study was also difficult. There a, is comparative study actually is very important for the betterment of mankind. Exchange of data among various scientists was also difficult because uh, people study biology all over the world, and if it is not possible to exchange their data, this becomes difficult to analyze anything for the betterment of biology and human beings. Then the organization, access, search, retrieval of biological information was thus difficult. Again, analysis and interpretation of biological data through computational approaches were impossible. Bioinformatics is a multidisciplinary approach. So, in late 1980s, various disciplines merged to give rise a new discipline which is known as bioinformatics. Disciplines that make up bioinformatics are as follows: quantum mechanics, electronics, genetics, artificial intelligence, biochemistry, molecular biology, mathematics, statistics, computer science, and information technology. These all merge to give rise to bioinformatics. So, there are various definitions also for bioinformatics because it is related to different fields of science, and person of persons of different fields define it according to their terminology. As for the history, a little bit history of bioinformatics. Discipline of bioinformatics with this name started between 1980 to 1990. HAL organized first international conference in 1987 and defined it as bioinformatics. This was a new subject of genetic data collection, analysis, and dissemination of to the research community. This can be defined in numerous ways, and investigators from different fields defined it differently. In whatever way it may be defined, its main goal is to find out secrets of biology for the benefit of mankind. The science seeks common patterns in biology, and its few definitions are written below. Application of computer technology to bioengineering is called bioinformatics. Application of information technology in biology is known as bioinformatics. What is .com defines it like bioinformatics is the science of developing computer databases and algorithms for the purpose of speeding up and enhancing biological research. Another definition is bioinformatics is the field of science in which biology, computer science, and information technology merge into single discipline. This was given by NCBI, National Council for Biotechnology Information. Now, another source says that bioinformatics is a process of developing tools and processes to quantify and collect data to study biological systems logically. BitsJournal.com says that bioinformatics is a combination of computer science, information technology, and genetics to determine and analyze genetic information. Bioinformatics is the use of computer tools to manage and analyze genomic and molecular biology data. Besides, bioinformatics is the use of computers in solving information problems in life sciences. Mainly, it involves the creation of extensive electronic database on genomes, protein sequences, etc. It also involves techniques such as three-dimensional modeling of biomolecules and biological systems. It was seen that during initial days of bioinformatics, word bioinformatics, computational biology, and informational infrastructure are or were sometimes used for similar meaning or were used exchangeably. So it is important to define them separately. In bioinformatics, databases are made, and so persistent data sets are stored in consistent state for infinite time. Algorithmic, algorithmic tools are used to understand biology in the discipline of computational biology, and bioinformational infrastructure is used to define all activities related to biology like maintenance of data, tools used to understand it, and networks used to exchange data. So, what are the aims of bioinformatics? Aims of bioinformatics are to organize data in such a way that researchers can access to data and they can submit new data. Also, whenever data is produced, data must be managed throughout its life cycle starting from the creation, initial management, archiving, or deleting. For example, gene bank receives data. Gene bank is a data bank which receives data of nucleic acid sequences and manages them in useful way. Second is uh, to develop tools and resources which can aid in the analysis of data. For example, BLAST and FASTA are the tools of bioinformatics which analyze sequences of macromolecules. Nowadays, tools are being developed to uh, study complete genome. And third is that uh, tools for data analysis are used and results are interpreted in meaningful way. For example, tools are used to perform global studies by taking more than one systems, and comparisons are made to find relations and new features among them. Functions of bioinformatics. So whatever are the aims, bioinformatics performs similar functions also. That is handling and management of large quantity of biological data, communication among researchers, access to data, search and retrieval of information, and computational approaches of databases. Branches of bioinformatics. Based on the type of material used for experimentation, bioinformatics can be divided into two main branches, which are as follows: animal bioinformatics and plant bioinformatics. Animal bioinformatics is the science that deals with proteomics, genomics, and metabolomics of various animal species. Help of computer is taken for this study. Topics included in the study are gene mapping, gene sequencing, animal breeds, animal genetic resources, etc. This can be further divided as mammal, reptiles, and bird bioinformatics, etc. Second branch is plant bioinformatics. Plant bioinformatics deals with computer-aided study of plant species. Studies are done on similar topics like gene mapping, gene sequencing, resources for plant studies, databases, etc. Here, agriculture bioinformatics or crop bioinformatics performs computer-aided studies on various agricultural crops. Horticultural bioinformatics is done for fruits, crops, vegetable crops, and flowers of various uh, flowers, crops. Medicinal plant bioinformatics deals with computer-based study of various medicinal plants, and forest plant bioinformatics includes studies of forest plant species. Apart from these two, microbial bioinformatics is another branch in which studies are done on various kinds of microbes. Opportunities and applications. There's various applications of bioinformatics, and numerous opportunities also. Trained and skilled bioinformaticists are needed by many bioinformatic in drug companies and for research and academic institutions. Trained people can identify genes, develop new gene products, can help in revolutionizing drug development and gene therapy, analysis of patterns of gene expression, and help to understand toxic responses and predict toxicity mm, of various drugs. Biocomputing provides the theoretical background and practical tools for scientists to explore proteins and DNA in many ways, like in sequence homology. What is sequence homology? In understanding the meaning of macromolecular sequences with two distinct analytical themes, which are as follows. In first approach, pattern recognition techniques are used to detect similarity between sequences and hence to infer related structures and functions. And in second approach, ab initio prediction methods are used to deduce 3D structures and ultimately to infer function directly from linear sequences. The direct prediction of protein three-dimensional structure from linear amino acid sequence is the objective of bioinformatics. One of the driving forces behind bioinformatics is the search for similarities between different biomolecules. Apart from enabling systematic organization of data, identification of protein homology has some direct practical uses. Theoretical models of proteins are usually based on experimentally solved structures of closed homologs. Wherever biochemical or structural data are lacking, studies could be carried out in yeast-like lower organisms, and the results can be applied to homologs in higher organisms, such as humans. It also simplifies the problem of understanding complex genomes. Analysis of simple organisms are done first, then applying these same principles to uh, more complicated ones solve the problems there also. This would result in identifying potential drug targets by checking homologs of essential microbial proteins also. Predictive functions. Through large-scale screening of data, one can address a number of evolutionary, biochemical, and biophysical questions. We can identify specific protein folds associated with certain phylogenetic groups, uh, common features among different folds within a particular organism, the degree of fold shared between related organisms, the extent of relatedness derived from traditional evolutionary tree, and the diversity of metabolic pathways in different organisms. One can also integrate data on protein functions, given that given the fact that particular protein folds are often related to specific biochemical functions. Combining expression information, structural and functional classification of proteins, one can predict the occurrence of protein fold in a genome, which is indicative of high expression levels. In conjunction with structural data, one can compile a map of all protein to protein interaction in an organisms. Now, medical areas. Application in medical sciences have centered on gene expression analysis. This usually involves compiling expression data for cells affected by different diseases and comparing the measurements against normal expression levels. Identification of genes that are expressed differently in affected cells provide a basis for explaining the cause of illness, and highlights potential drug targets. With this information, one can design compounds that bind to the expressed protein. Given a lead compound, microarray experiments can be used to evaluate responses to pharmacological interventions. It can also help in providing early tests to detect or predict the toxicity of trial drugs. If bioinformatics is combined with experimental genomics, a lot of advances could be made to revolutionize the future healthcare programs. This involves postnatal genotyping to assess susceptibility to or immunity for specific diseases and pathogens, prescription of a unique combination of vaccines, minimizing the healthcare costs of unnecessary treatments, and anticipating the onslaught of disease later in life, which could lead to guidance for nutrition intake and early detection of any illness. In addition, drug-based treatments and anticipating the onslaught of disease, which could lead to guidance for nutrition intake and early detection of any illness. In addition, drug-based treatment could be tailored specifically to the patient and disease, thus providing the most effective cure of medication with minimal side effects. Race-based BIDL may be first personalized medicine in future, which is a medicine for heart failure. It was developed or because it shows great reduction in mortality rates from heart diseases among African Americans. The compound, however, has not been found to work well in Caucasians. Primer designing. To get a lot of DNA molecules from sorry, various purposes, we run polymerase chain reaction, and for that we need primer. Bioinformatics helps in getting primers for the reaction if the sequence of DNA is known. Now, what is reverse vaccinology? Many immunoinformatics informatics algorithms and resources have been developed to predict T cell and B cell immune epitopes for epitope vaccine development and protective immunity analysis. Similarly, protein candidates for vaccines are predictable in silico from genome sequences using reverse vaccinology. Key element of reverse vaccinology is bioinformatics. Human Genome Project has benefited forensic science, pharma industry, discovery of beneficial and harmful genes, contributed to a better understanding of human evolution, diagnosis of disease and disease risk, genetics of response and therapy, and customized treatment, identification of drug targets, and gene application. There are many more applications of bioinformatics, starting from preparation of databases for nucleic acid sequences, for protein sequences, for fatty acid sequences, for metabolism, for uh, various compounds which can be used for drugs, for various candidate vaccine compounds, and whatnot. They are used in bioinformatics is used in hybrid technologies. It can be used for making biosensors. Animal tissue engineering technology also utilizes the knowledge analyzed from bioinformatics. DNA chip technology needs primers, so which can be if DNA sequences are known, primers can be made, gene expression can be done, measurement of biodiversity can be analyzed with the use of bioinformatics. Um, Total genomic complement of one particular environment from all species present define the biodiversity of that ecosystem. Data from all species are stored in databases. This helps in studying population dynamics, calculate cumulative genetic health of a breed pool in agriculture or endangered population in conservation. Phylogenetics can be studied by studying sequence homologies, microbial gene applications. We can use microbes for energy production, environmental cleanup, industrial processing, and toxic waste reduction. And for that, we must know the sequence or genomes of microorganisms and manipulate them for our use. So, for that, comparative studies are done. In veterinary science, uh, by studying genomes of farm animals like cows, sheep, etc., it becomes very easy to improve production of livestock and ultimately have benefits for human nutrition. Same is true for crop improvement. We can produce drought resistant varieties and improve the nutritional qualities of crops. We can develop the insect resistance in plants. Besides, evolutionary studies can be done. We can see how the amoeba works and how human beings uh, or primate cells of primate work, and what are the similarities and what are the differences. For that matter, I can say that trypsin enzyme is present in amoeba also, and in primates, higher animals also. So that is a preserved protein throughout the evolution. Bioinformatics can be used to in field of forensic sciences. Forensic analysis of microbes can be done. This already helps to know exactly which bacteria is present at the crime site or used as bioweapon. Antibiotic resistance in microbes can be studied with the help of bioinformatics. Then interactome. The interaction is the sum of all proteins interactions in cell, and this is represented as graph with proteins or nodes and interactions as links. Each two hybrid system produces large amount of protein interaction data. When large number of proteins are considered for making maps, they become too complex, and then clustering of functionally similar protein is done to make functional interaction map which links fundamental cellular processes. So bioinformatics has uh, innumerable uses in our life. Thus, to find out these, we can define the fields and areas of research in bioinformatics. Irrespective of the branch of bioinformatics, every branch deals with following fields. There is immense possibility of doing research in all these fields, like in the field of structural bioinformatics, which analyzes the structures of macromolecules, drug designing, which compound can act as a drug, and its interaction with the cellular components, phylogenetics, to find out the find out the role of evolution in the development of different organisms, computational biology, population genetics, genotyping can be done, splice site prediction, like the splice site means in RNA, pre-messenger RNA when matures into uh, mature RNA, splicing occurs. So, at which site splicing is occurring can we find out. miRNA prediction can be done. RNA structure can be predicted. Gene can be predicted. Gene structure, its organization can be predicted. Transcription occurs in DNA. So, which element acts as transcription binding site? Genome annotations can be done. Again, that shows the structure and function of various genes present in gene in genome. Ancestry can be predicted. Mathematical modeling is required to find out the structures or solve the structures. Uh, ethnicity predictions can be done. Functional domain predictions can be done. Various proteins have, though they are not similar yet, they contain similar domains with similar function. So they various proteins, a part of various proteins can perform similar function. They can be found out. Motif also serves similar function in the structure of proteins and nucleic acids. These patterns can be matched and their functions can be predicted with the help of protein-protein interaction. The various functions can be found out. Protein folds while coming after it is made in the cell, it is folded in some particular manner, which could be find out. All the data can be then stored in the form of databases, and then the one field can be development of databases. According to data which we get after analysis, we can make databases for its storage and handling forever. So computers have a very important role in bioinformatics. Sorry. Mm. Bioinformatics is largely, although not exclusively, a computer-based discipline. It utilizes advances in the area of computer science, information science, computer and information technology, and communication technology to understand complex problems of life. At present, advancements in information technology, particularly the internet, are being used to collect, store, and access ever increasing information in biology. Role of computers in bioinformatics is To understand biology through computers, it is necessary that computer recognizes biological language. Computers use binary language represented by 0101, which is similar to alphabetical language used in organic molecular structures. For example, AUGC for 4 nucleotides, which are basic units of nucleic acids, DNA and RNA, and AGLMN for 20 amino acids, which are basic units of proteins. DNA and protein structures are strings of basic units, and these alphabetical strings can be stored in the form of binary language. Thus, biomolecules can be represented in a language that machine understands, and it becomes possible to use computer to solve biological problems. In order to understand biological problems, same task is required to be repeated several times, and it is easy to do this with the help of computers instead of doing manually, because in computers data can be stored till the completion of the study, and inferences can be curated, that is in the form of database. Data is handled throughout its life cycle. Since databases are storehouses for knowledge, it is possible to retrieve data from them and used for user himself, or may be communicated to other users. Various algorithms may be written for biological problems. Tools may be developed by using them, and problems may be solved for example, using computer programs. It is possible to express various patterns in biological molecules, in graphical representations, visualization of structures of macromolecules, understand molecular modeling by performing docking between two structures, and stimulate the docking processes, etc. These are the various references which I used for preparing my lecture, and thank you for today.